All right, thanks very much, Joel, for the introduction, and welcome everyone to this webinar. So my name is Yannick Bussweiler. I'm an application scientist here at Tofware for laser ablation, and I'm actually a geologist by training, so I apologize in advance that the examples today will be biased a little bit towards geological applications, but I will also try to add some biological examples as well. So yeah, the title of today's webinar is Capabilities of the icp-TOF for Laser Ablation Applications. So this webinar can be divided into three main parts. First, I will discuss the instrumentation. So I will describe the icp-TOF and also talk about the development of fast washout ablation cells. And these are basically the technical requirements we need for fast multi-element imaging. And here I will talk about one example, a geological example, namely imaging of the mineral garnet. And then I'll also add a couple of biological examples. And then the last part will deal with how we can speed up routine laser ICP-MS spot analyses with the combination of using the icp-TOF and these fast washout laser ablation cells. Okay, so first of all, what is laser ICP-MS? I guess that most of you are familiar with this technique, but it's here just a quick introduction. So it's when we use a pulsed laser beam to sample a solid sample. So this can be a mineral or also biological samples like tissue samples. And then we have a carrier gas that transports the aerosol, the sample aerosol, out of this ablation cell via some tubing. And this will go into the inductively coupled plasma, so that's the ICP part. And this basically ionizes all of the sample. And the stream of ions can then be analyzed in a mass spectrometer, so a mass overcharge analyzer. And the main advantages of this technique of laser ICP-MS is that you have fairly high spatial resolution. So with modern laser ablation systems, you can go down to 1 micron in laser spot size. And it's also a good method if you want to measure a lot of elements at once. So you can measure major, minor, and trace elements together. So it has a good linear dynamic range. Okay, but here we're talking about time of flight mass spectrometry. And this on the left here is our icp-TOF instrument. It's about the size of a fridge, a European-sized fridge, I should say. So it's fairly compact. And some of you may notice that we use the ICAP-RQ, which is a quadrupole instrument from Thermo. And we add a time of flight analyzer here on the top. So in the schematic view here in the middle, you can see that we actually keep the original ICP source. We keep the ion optics. We also leave in this Q cell, which is a reaction or a collision cell, if you want to get rid of certain interferences. And then here, this part, that's where the original quadrupole mass analyzer would have been and the detector. So we take that out and we replace it with a notch filter. This can be used to attenuate certain ions. For example, argon ions that come from the plasma, you can attenuate those so that they don't hit the detector all the time. And then here at the top, you see the time of flight analyzer, which we built here at Tofware. And I will explain quickly how this works. So over here in the schematic view, the main difference to sequential mass spectrometers like quadrupole or sector field instruments is that we actually introduce all of the ions together. So they all enter this extraction area here where they're accelerated to equal energy. So this happens via pulsed voltage. And because they all have different mass and the same energy, they must fly at different speeds. So in this scheme, the lighter ions travel faster than the heavier ones. And then at the detector here, this is basically what you see. You record intensity in millivolts against time of flight. And if you know the identity of some of these masses, you can perform a mass calibration and you can then transform this time of flight axis into mass overcharge. So the unit here is Thomson. Yeah, the idea is that this happens all very quickly. So this pulsed extraction happens at a frequency of 33 kilohertz. And that means that you actually record 33,000 mass spectra per second. And if you want to read up on the specifications of the icp-TOF, there's 2 recent papers. So Hendricks et al. Described the system in solution mode, and Berger et al. Describe it in laser ablation mode. All right, now let's talk about the fast washout laser ablation cells. So this is actually something that a lot of laser ablation companies work on these days, not just Teledyne CTEC, but also other companies like ESI, New Wave, or Australian Scientific Instruments, ASI. But I'm just showing the Teledyne CTEC equipment here because that's what we have in the lab, and most of the examples that I will show use this equipment. So here we use this Analyte G2 laser, which is an argon fluoride excimer laser, and has this wavelength of 193 nanometers. And that is good for ablating solid samples like minerals, but also biological samples. And then this is equipped with this Helix 2 sample cell. And this is a dual volume cell. That means we have this ablation cup here in the center. And this basically helps to keep all the aerosol in one place and transport it efficiently out of the cell. And it is also equipped with this aerosol rapid introduction system, ARIS in short. And here you can see that the tubing is thinner than in conventional systems. So by reducing the inner diameter of this tubing, you can already speed up the aerosol transport. And then you can also mix in extra argon here, which also speeds up the aerosol transport into the ICP. Yeah, and the main advantages that we get from these fast washout cells, that we have reduced aerosol dispersion. So that means we get a nicely focused signal and also better signal to noise ratios. And this basically enables us to do spot resolved imaging. And again, if you want to read up on these recent developments of fast washout ablation cells, you can check out these references down here. Here at Tofware, we use a triggered approach to do this spot resolved imaging. So that means when you fire a shot with the laser here on the sample, at the same time we send a trigger signal to the mass analyzer, so to the icpTOF. And here you have to take into account the so-called aerosol transfer delay. That's the time it takes for the aerosol to travel from the laser ablation cell through the tubing, through the ICP, and all the way to the detector. And this delay time is dependent on the sample material, because different materials have different ablation behaviors. And of course, it also depends on the ablation parameters that you're using, so spot size and laser energy. So that means that you have to tune this aerosol transfer delay. And we're currently working on a control software that does this automatically. It's called TOF Pilot. And here I'm showing a screenshot from this TOF Pilot software. And you can see here the signal from a single laser shot. And you can also see the aerosol transfer delay, so the time it takes to travel to the detector. And this can be shifted so that the signal always arrives within an integration window. And at the same time, the width of this signal from a single laser shot defines how fast you can run the laser, so the laser repetition rate. So in this example here, the width is about 20 milliseconds. That means you can still run the laser at 50 hertz without getting too much overlap. Yeah, and this precise triggering approach for imaging is described in our article in Spectroscopy. All right, so that was it for the technical part. Now we'll talk about the first application example, which is high-speed, high-spatial resolution, multi-element imaging. So as I said, this will be a geological example. So here we're looking at a garnet crystal, which was kindly provided by Gabriel Sabau at the Geological Institute of Romania. And this garnet grain is about 5 by 7 millimeters in size. And he already had some element distribution maps for this particular grain. So over here you can see maps that were recorded with the electron microprobe, which is a popular instrument amongst geologists. And they produce these nice images, but only for major and minor elements. And this also takes quite a long time. So this image here was recorded in something like 10 hours. So we wanted to test if we can speed this up using the icpTOF. And again, for this, we used our TOF Pilot software. Now, I will just quickly walk you through the workflow that we use here in TOFPilot. So it all starts on the laser computer, where we set up, first of all, the ablation parameters that we want to use. So the fluence, the repetition rate, and the spot size. And then we draw the image as a raster of side-by-side spots here on the sample. Then in the TOFPilot software, you can download all this image information, so the XY coordinates and so on, and this will then recreate a grid. And then you can start the ablation from TOFPilot. Another really nice thing is that you can watch how the image is constructed in real time. So basically, you watch how each pixel is filled with data from the icpTOF. And while this is happening, you can flick through different isotope channels, and you can change the display settings and stuff like that. And then here schematically, you can see how the image is constructed over time. And this is really nice because you can see if something goes wrong, so you don't lose valuable sample material or analytical time. And then keep in mind that in the final image, each pixel actually contains not just one element, but the full mass spectrum. And of course, this can then be exported for further data processing. And all of this is described in our white paper, which you can find here on our website. All right, so here are the results. So as I said, we use the Analyte G2 laser for this, equipped with the ARIS device for fast washout. And in this example, we're actually able to run the laser at 100 Hertz because the signal width of the single laser shot was just 10 milliseconds or so. We used a spot size of 20 microns, so fairly high spatial resolution. And this was the sort of fluence we used. And again, these are spot resolved images, so each laser shot gives you one pixel with the full elemental information. And first of all, what you can see here is that we could reproduce the EPMA element distribution maps in far less time, so this took less than 1 hour. And at the same time, we get information for all other elements. And what was really interesting for this particular grain is the rare earth elements, because they show this nice zonation with heavier rare earth elements being concentrated in the core of this grain. But these are just intensity images. So what do you have to do in order to quantify them? I guess there's one additional step that you don't have in conventional laser ICP-MS. And this has to do with the fact that we actually introduce all the ions at once into the mass analyzer. So we get a characteristic TOF baseline. So you can see what this baseline looks like over here. Just keep in mind that this is a log plot, so it's actually not quite as bad as it looks. And we also have software. Now in this case, this is called Tofware, that we can use to characterize these TOF baselines and then subtract them. So in general, Tofware can be used to, first of all, check the mass calibration, then to subtract the baseline. And you can also do fancier things like peak deconvolution if you're worried about certain interferences. And all in all, this provides an efficient way of processing large and complex data sets that we collect with the icpTOF. And then from here, you can export the process data, for example, as CSV files. And these can then be loaded into third-party software like iLite, which geologists like to use for quantification. So this is the reference here for iLite. And yeah, the way this works, you have basically the data for your image, and then you've also measured some calibration materials before and at the end of the session. And you also measured gas blanks. So first you would subtract the gas blanks from the image. Then you normalize everything to an internal standard, and then you set everything in relation to the calibration material. So these are then the quantified results. Here I'm just showing that we can quantify everything from major elements down to trace elements. So calcium, tens of weight percents, yttrium, a few hundred ppm, and lutetium over here in the single ppm level. Okay, then there's a recently published add-on for iLite called Monocle. This is the reference here. And this actually gives you a flexible tool of interrogating these element maps. So for example, you can define regions of interest, and you can get real-time information about certain areas. For example, here, this gives you a histogram of the concentration of manganese in this case. But you can also create normalized rare earth element patterns in real time with the software. So this is particularly useful for geologists, but I think it may also be applicable to other samples. So now let's look at a couple of biological examples of fast imaging. So one is this nice paper here by the University of Ghent group, Christine van Malderen et al. And here they looked at sections of Daphnia, which is a water flea. So they produced 59 sections of this specimen. And then each section was imaged by LA-ICP-TOFMS. And in this case, they used a 5-micron laser spot and a repetition rate of 20 Hertz. So each section took about 12 minutes to image in multi-element space. And what they did next, because they already had information from CT, they could basically realign all these sections and then produce a 3D image of this water flea that shows the distribution of certain trace elements. So in this case, copper, zinc, and nickel. Another biological example that we did here in-house at Tofware was in collaboration with the University of Copenhagen. So they brought some sections of plant roots, and they were interested in the distribution of certain nutrients within these roots. And because they were so small, we had to use a very small laser spot size, here just 3 microns. And again, we used a repetition rate of 20 Hertz to do the spot resolved imaging. And this is basically what we got. You can read up on this on our website again. That's where you will find the application note. All right, so that was it for the high-speed imaging. Now let's look at how we can speed up normal spot analyses by using the icpTOF in combination with these fast washout cells. And this example that I will show you here is a collaboration with Stefan Poitras and Graham Pearson at the University of Alberta. They provided us again with samples of garnet. And in this case, these were exploration samples that were collected here all the way in northern Canada. Okay, so over here. And they mounted the garnet grains in these epoxy pucks that were then polished and carbon coated so that they could, again, use the electron microprobe. And yeah, EPMA was used to get the major and minor elements. And here, this already took about 1 minute per grain. And then in order to get at the trace elements, they did routine laser ICP-MS. In this case, they used a sector field mass spectrometer. They used 90 micron spots, repetition rate of 10 Hertz, so that the analysis took about 2 minutes almost. And the way this was done, first, they recorded 30 seconds of background, then they ablated on the sample for 40 seconds, so using the 10 Hertz repetition rate. And then they had to wait for another 40 seconds for all the sample to be washed out of the cell. So all in all, you have over 3 minutes for full characterization of one garnet grain. So I guess our objective was to test if we can speed this up with the icpTOF and these fast washout laser ablation cells. So again, we use the Analyte G2 in combination with the ARIS device. And we set the icpTOF to continuous acquisition mode, so there was no triggering. And we also set it to 100 millisecond integration time. So that means that 3,300 complete spectra are averaged into one. So with this setup, we could then perform laser ICP-MS compressed in time and space. So that means we could use smaller spot sizes. And we also ablated for a shorter time, but at a higher repetition rate. So it would look like this. In the background here, you see the conventional approach. And here in black, our new speedy approach. And you can see that we can collect 9 times as many samples in the same time. So if you take into account the backgrounds and maybe doing 3 replicates per grain, this would take about 20 seconds for full characterization of one garnet grain. So here are the results. I've used the NIST 612 glass for calibration. Again, this was done with iolite. And here I'm just showing all the elements that I could quantify from silicon down to thorium, just sorted by their concentration. And I've also plotted the limits of detection as calculated by iolite. And you can see that limits of detection are down to PPT levels here for thorium. And yeah, all the elements were above LODs. Then we can compare the results first to the EPMA data, so the data from the electron microprobe. And here I'm basically assuming that the EPMA data are accurate to within 10%. And you can see that the TOF data basically fall along this range, so they're accurate to within 10%. One exception here is magnesium. And here I think we're actually looking at a calibration issue, because in the NIST 612 glass, we only have 40 ppm of magnesium or so. And in the sample that we're trying to quantify, we have tens of weight percent. So I guess if you used more appropriate external calibration material, you might also be able to quantify the major elements. Now comparing the TOF data to the sector field data in terms of trace elements. Again, I'm assuming that the original LA-ICP-MS data are accurate to within 10%. And you can see that the ICP-TOF data fall within 10% for all the trace elements. And then when you plot the trace element data, so for example, that one thing that is popular amongst geologists is to plot these normalized rare earth element patterns. So for the garnets, you then see different groups. So the first group here, shown by the black patterns, is this normal pattern. Then we see a more sinusoidal pattern in blue, and another pattern in red, which is depleted in the light rare earth elements. So this is the original sector field data, and here's our ICP-TOF data, and you can see that the patterns are basically identical. And keep in mind that over here, this only took 20 seconds per pattern, and this in the conventional laser ICP-MS approach takes 110 seconds. Another element that is important in garnet studies, specifically in mantle garnet studies, is nickel, because this can be used for geothermometry. That means you can calculate temperatures from the nickel concentrations. But for this, you have to know the nickel concentration quite accurately and precisely. So here I'm plotting the TOF data directly against the sector field data. And again, you can see that they fall along this one-to-one line to within 10%. And I should also mention that we're looking at different uncertainties here. So in the TOF data, I actually used the 2 standard deviations from 3 replicate measurements on each grain. And over here, the original sector field data only had the standard error from iolite with it. So this is more an inter-spot variability rather than an intra-grain variability. And one thing we noticed is that some samples had more variability in the nickel concentration. And here we went back to the original ablation signal. And sure enough, we found spikes in nickel in these samples. And first we suspected that these could be nickel sulfides, which are common in mantle garnets. But then the nice thing about the ICP-TOF is that it records all the elements together. So you can actually check the sulfur signal. And there was no spike in sulfur. Instead, we saw a correlation with cobalt and platinum here. So these are more likely to be alloys. So that's pretty nice because you can go back to your samples and basically characterize micro-inclusions. And that brings me briefly to one more application area that I can't talk about in detail here, but it's the analysis of fluid inclusions. And there was a nice paper here by Mathieu Hallot et al. 2015, where they compared capabilities of sequential, so that's quadrupole or sector field mass spectrometers, against quasi-simultaneous laser ICP-MS, so using the ICP-TOF. And they found that, especially for small inclusions, so smaller than 10 microns or so, there are benefits to using the TOF, which is shown here by lower detection limits for all these elements. So in summary, I hope that I was able to show you that the combination of the ICP-TOF And these new generation fast washout laser ablation cells give you new capabilities. So for example, you can do faster multi-element imaging, and you can basically perform spot resolved multi-element imaging. That means that each laser shot gives you one pixel in the image, which has the complete mass spectrum information in it. So there's no need to pre-select analytes or isotopes. And this can be nice because you might find things that you didn't even expect in your sample. And the repetition rate is basically defined by the signal width that comes from a single laser shot. And in this Garnet example that I showed you, we could use 100 Hertz. But as far as the ICP-TOF is concerned, this is not the limit. We're so far limited by the washout time. Then the accelerated spot analyses. And this one example that I showed you on the mantle garnets, we can basically conclude that we only need one method to characterize the full spectrum of elements of interest. So we don't even need the electron microprobe. And this then saves you a lot of time. And if you compare the method of LA-ICP-TOFMS directly to conventional LA-ICP-MS, we had an increase of, or we were a factor of 9 faster, so that you can actually analyze 180 grains per hour instead of just 20. And I think we had good precision and accuracy for major, minor, and trace elements. And because we also have full elemental information, you can also go back and identify micro-inclusions in your samples that perhaps you did not expect. Okay, so we want to thank our collaborators here, all these research institutes and universities, for past and ongoing collaborations. We also want to thank the laser ablation companies, Teledyne CTEC and ESI New Wave for collaborations, and the developers of the iLAD software and the ACE Nano project. And as always, please don't hesitate to contact us if you have any questions or ideas for collaborations. So this is Martin Tanner, the product manager of the ICP-TOF. Then there's Olga Borovinskaya, who's an application specialist for solution and single particle analysis with the ICP-TOF, but she also knows a lot about laser ablation. And then there's me, application scientist for laser ablation and geological applications. So here are the references of this webinar. Thanks very much for listening.